Hello, my name is Claude Robert, and I'm the Director of Platform Development within EmbryoGENE, which is a national research center from Canada, which is supported by INSERC. Within this research network, we have the mandate to define embryonic health to the study of bovine and porcine embryos submitted to different environmental stresses, namely from the application of assisted reproductive technologies or from maternal nutrition. Within EmbryoGENE, two transcriptomic platforms have been developed to study the porcine and the bovine embryonic transcriptome, and we also developed a platform to study DNA methylation in bovine embryos. So within this presentation, I will talk about the development of the epigenetic platform. So within the topic of research of interest to EmbryoGENE, we're also interested in understanding what makes an embryo and what defines the embryonic developmental competence and also embryonic quality. And as mentioned, the mandate is to better understand how early embryo cope with stresses. So to develop a platform dedicated to the study of DNA methylation, we have to take into consideration the content of the DNA content within a bovine embryo. So it's a large mammal, so it's about 3 billion base pair. In each cell, there's about 6 picogram of DNA, and a blastocyst is roughly composed of 100 or 180 cells. So if we want to take into consideration that most current DNA methylation platform necessitate about 1 or 2 microgram of DNA, it's a lot of blastocysts to collect. So we decided that we would like to develop a platform that would necessitate an input of about 10 nanogram of DNA, which correspond more or less to 10 blastocysts. So we started by surveying the options that were out there. So right here, what we can see is that there is a pre-treatment uh, step that is necessitated to be done to highlight the methylation. So it's either based on restriction enzymes, which use enzymes that are sensitive to methylation and will not cut in the presence of methylation. The well-known bisulfite treatment, which converts unmethylated cytosines into uracil, or the affinity-based systems which pull down the methylated cytosines. Once these samples have been treated, then it's necessary to identify the regions of interest either by candidate approach using PCR or sequencing, or by profiling the entire genome using different means. So using these options, we decided that we wanted to use a platform that was similar to the transcriptomic platforms. So we based our approach on restriction enzymes combined with a microarray to identify the sequences. So this came along with our survey of the literature, where we found that a lot of the techniques out there are requiring a lot of starting material, which is impractical for early embryos. One of the method that is currently very acceptable is the reduced representation bisulfite sequencing, which seems to work with about 30 nanograms of starting material. Another question we had to answer was where was the DNA methylation in early embryos? So is it in the CPG islands right here, in the promoter regions, or is it in the gene body, or in enhancer or silencer elements, or maybe very upstream in intergenic elements? So we decided to use a unbiased surveying method to identify the regions where the methylation was in early embryos. So it's I won't go through this, but it's basically it's a subtractive approach that is used to pull down or enrich a soup in different in regions that are methylated within the genome, and then we sequence the soup to identify the potential target loci. So what we found using three different approaches was that the not only the promoter regions were methylated, but the gene body accounted for a large proportion of the methylation and the intergenic regions. So we need to address these questions within the development of the platforms. What we also found is that we use different pairs of enzymes that were either specific to methylated cytosins or hydroxymethylated cytosins. And what we found was that different repeated elements, either the sign or the lines, were more bearing marks of hydroxymethylation, while satellites were bearing the standard the methylated cytosine marks. This was the profiling that different repeated elements was um, bearing different methylation mark was supported last year by a nice publication from Alexander Meissner. So right here, what you have is a schematic of the development of the platform. So the first step was to find the loci. So find the region where in the genome where the methylation was established in early embryo. So we, as mentioned, we did that using the subtractive approach. And then we needed to develop a sample preparation strategies that would work with only 10 nanograms of starting material. We needed to test the platform and define the data analysis pipeline that would work with this platform. So for the sample preparation protocol, we tested a few of them and we decided to go with with this one, which fragments the DNA using the MSC1 enzyme that does not target any cytosins. So this is only to shorten the DNA. And then we add linkers to these fragments, and then we proceed to the methyl sensitive enzymes, which are going to cut in the restriction enzyme that is not protected by methylation, and will leave intact the sites that are methylated. So by proceeding to PCR amplification, what we do amplify are methylated regions right here. A question that we asked was when we cut the genome with MSC1, what is the size, the average size of the fragments? And you can see right here that they're pretty small, so about 100 base pairs. So and very little fragments are very long, so longer than 2.5 kbs. So which means that we don't see much of these fragments that have a lot of restriction enzyme sites within the same fragments. Right here, it's an example of the tiling that was done to the DNA to produce a DNA methylation array. So right here we have a genomic region, and you can find right here in the region that we tiled the CPG islands. So we found in silico all the CPG islands, and there's an oligo on the methylation array that is tiling all the CPGs found in the bovine genome. And right here are the cuts done by the MSC1 enzyme, and we tiled most of these or many of these fragments. Right here, it's the oligo that is used on the transcriptomic platform to target this gene. So we start with an initial version of the microarray containing 1 million oligos that we tested, and then we decided to go with a version of with a smaller version of the microarray containing 400,000 oligos. And the reason for this was to reduce the cost. So we can put two of these 400K array on a single slide, which divides the cost by two. And we did a second version of this microarray, which was a selected probe or selected population of probes from this 1 million oligos. So all these probes are known to be good and to be generating fluorescence intensities. What we noticed with the first version is that some of the probes never generated any signals, and what we believe is happening is that within some of these fragments there are very difficult template to amplify. So these will never generate any signals. So instead of, let's say, for example, putting a probe right here that will never be highlighted on the microarray, we decided to move this probe elsewhere, let's say right here on the genome, to replace it by a functional probe. For the purpose of quality control, what we also included in the platform are pairs of spikes. So you can see that for each pair we have one spike that is containing the restriction enzyme sites that has been methylated using in vitro using bacterial DNA methylase, and another one that is not methylated. So that one should be digested with the restricted enzyme sensitive to methylation, but this one will not. And all these pairs also contains the MSC1 site, which is a control for the initial fragmentation of the DNA. So the samples are processed, and then we test by qPCR the extent of digestion, and what we're looking for is that 5-cycle difference between the positive or the methylated, the protected fragments compared to the unprotected fragments in the samples. So right here, what you have in the table is the percentage of digestion that is associated with the 5 cycle difference between the two types of spikes. And so all samples that we use are at least 97% digested. So this is important to limit the false positive. If, for example, a sample at this point does not have the minimal number of 5 cycles threshold, what we do is we add enzymes and we process the samples for further digestion to make sure that the samples are fully digested. We have within the data analysis pipeline, and we have on the microarrays probe that account for these spikes. And right here we can see what happens when you're facing a high quality sample. So this is the fluorescence intensities for the probes on the methylated fragments. So they generated fluorescence, and for the spike that was not methylated, you can see that the intensities of the probes are very, very low. When you have low quality samples, you can see that the digestion was not complete and these probes are generated signals. So having these three different steps done, what we decided to do was to test the DNA methylation platform. So what we do is we take embryos that we from control embryos, control treatments or treated, and from the same pools of samples we can both extract the RNA and the DNA. The RNA is submitted to the transcriptomic platform, which we call EMBV3, and the DNA is submitted to the DNA methylation platform, which we call EDMA, for EmbryoGENE DNA methylation array. So then comes the complex data analysis. Right here you have the transcriptomic data analysis that is used. So it's done using a standard platforms or standard algorithms to generate the differentially expressed genes, and then we can further bind this information, taking into account the mRNAs or the long non-coding RNAs. Sorry. For DNA methylation, well, it's a bit more complex, where we have to account for genes, for repeated elements, for probes that are within the gene bodies or within the CpG islands. So CpG islands are classified according to densities or length, and also the genomic position. So using these different clusters or different binning strategies, we hope to define different or make sense of the DNA methylation survey. So these are examples of output that are generated from the platform. So right here, when we account for CpG island density, this is what the normal genome represent, and we look for enrichments or in certain types of elements. So right here we would have a difference in CpG islands of high density and of intermediate density. There's also impacts on the length right here, and the genomic position, if it's directly in the CpG islands or in the, and we don't see any difference in the shore, the shelf, or in the open sea. So right here we can see differences if in the gene body, and right here it was methylation that was observed within the exonic portion of the gene body. We also treat the repeated element, and depending on the classes, if there are sign, line, LTRs, simple repeat, and low complexity element. And finally, what we do is try to link the transcriptome and the DNA methylation profiles by comparing the right here you have the differences in between the treatment in transcriptomic information, and right here you have the information or the differences in the DNA methylation information, and this is positioned according to the over the entire genome. Right here you can see the chromosomes and the binding of the chromosomes, and what we're trying to see if there's a fit or a match or a correlation between the DNA methylation information and the transcriptomic information. So in conclusion, here we have a platform that works with low DNA input, so with pools of 10 blastocysts. It's adaptable and flexible, so we can ask questions regarding 5-methylcytosine, 5-hydroxymethylcytosine. The microarray can be used to identify fragments or loci that have been pulled down. It can be used on different experimental designs, so to see the DMRs, the differentially methylated regions found between in relation to treatments, or we can also profile the entire methylome using that platform. It's capable of handling both the transcriptome and the methylome from the same sample. That's important. And it comes with a full data analysis pipeline that brings the users from generating the data to trying to or making sense of the data using the different binning strategies. So I would like to acknowledge the people behind the platform. So Dr. Beatriz de Montera, who worked at the clone collection, finding out where the methylation was in the bovine early embryos. Dr. Jason Grant, who's working on the limbs and all the data analysis, not the pipeline, but the limbs, the recording of the treatment information or the sample information. And he also worked on identification of the CpG islands in the bovine genome. Dominique Gagne, who worked on the sample preparation procedures with right here a PhD student, Habib Shojaei, and Eric Fournier, who's working on the data analysis pipeline per se. So anything from the QC modules to the data analysis, the downstream data analysis. So I'd like to thank you for your attention, and if you're interested or have any question or comments, you can contact me at this email address or contact any of my colleagues, either Dr. Marc-André Sirard or Dr. Julie Minen, who are part of the EmbryoGENE network, and you can contact us for any questions. So don't hesitate. Thank you very much. Bye bye.